Good morning. Today my topic is sequence alignment. What is sequence alignment? Sequence alignment, it is a procedure which is mainly used to identify the structure, function and the evolutionary relationship between two sequences. Sequence alignments are two types. There are two types of the sequence alignment. First of all, pairwise sequence alignment, and the second is multiple sequence alignment. Pairwise sequence alignments are also divided into two types of the alignment: local alignment and the global alignment. What is pairwise sequence alignment? Pairwise sequence alignment, it is a type of the alignment in which we can align only two sequences at a single time. Local alignments or global alignments. Local alignments are the alignment which is used to align the highly conserved region between two sequences. It is used to calculate the maximum matches and the highly conserved region in two sequences. For the example, here I have two amino acid sequence, two nucleic acid sequences, adenine, guanine, thymine, cytosine, and we are compare the sequences with the next sequences, and we find out that there is 3 nucleotides which are highly conserved, and the others are maybe it can maybe matches, mismatches, or the gap penalties. And the second is global alignment. Global alignment, it is a type of the sequence alignment. It's aligned the entire sequences at a time. For the pairwise sequence alignment, this is the example of global sequence alignment. Here we are aligns two sequences, adenine, these are also the nucleotide sequences, but here we are aligned the all sequence till the end. It find out the maximum number of matches, mismatches, and the gap penalties. For the local alignment, we use Smith-Waterman algorithm. It is based on the dynamic programming algorithm, and for the global alignment we are using Needleman-Wunsch algorithm. It is also the dynamic programming based algorithm. Multiple sequence alignment, and the other methods for the pairwise sequence alignment is the dot matrix method, dynamic programming methods, and the K-tuple method. K-tuple method, it is also called the world method. K-tuple method, it is based on the BLAST and the FASTA program. What is BLAST? BLAST, it is the basic local alignment search tool which is mainly used for align two sequences. It can be protein sequence and other nucleotide sequence. And the dynamic programming method, it is based on the algorithm Needleman-Wunsch algorithm and the Smith-Waterman algorithm. Dot matrix methods for the local elements, it provides the graph. On the basis of the similarities between two sequences. This is in this way it provides the graphs. Maybe it is a graph between the lambda phage and P22. It's also bacteria. Here we are producing a graph 0, 50, 100, 150, 20, 50, 100, 150 or 20. So on the basis of similarities, maybe this reason represent the highly similarities between two bacteria. On the basis of we are creating this dot matrix graph. It is also the part of the local alignments, pairwise sequence alignments. Multiple sequence alignment. Multiple sequence alignments is the type of the alignments where we can align more than two sequences at a time. Multiple sequence alignment, it is the by which we can find out the highly conserved region between two sequences at a time. Yeah, between more than two sequences we can find out the highly conserved region. It is the extension of the pairwise sequence alignment, and here we are using for the methods for the multiple sequence alignment. These are also dynamic programming methods and dot matrix methods, iterative methods, which is used for the multiple sequence alignment. It is most sophisticated than the pairwise sequence alignment. Why we are using sequence alignment? Sequence alignment we are mainly used for the homology modeling. By the homology modeling, we can identify the ancestor of the sequence. If we have two sequences which are closely related to the species, on the basis of the alignment of those sequences, we can find out the ancestor of those sequences, and on the basis of the similarities of that sequence. And what will be the issue for the sequence alignment? We are required the equal number of nucleotides or the amino acid sequence for the sequence alignments, and we are also required for maximum number of the matches or mismatches in the sequence alignment. Multiple sequence alignment it requires the more sophisticated methods for in compared to the pairwise sequence alignments. We are saying that there are many methods for the multiple sequence alignment, iterative methods, dynamic programming, progressive methods. and the motif foldings. What is motif? Motif, it is the secondary structures of any nucleotide protein या फिर any amino acid sequence. It have the maximum conserved region by which we can find out that how many proteins are similar, dissimilar, and how many similarities are present between the two sequences. And on the basis of motif, we can create the tertiary structure of the proteins, which is called the domains. In the tertiary structures of the proteins, it have its self folding, which is called the domain, and by domain we can create a new proteins, which is we can also say this as a domain. It is the individual proteins, and it defines the all characters of the all important characters of the protein. Overall, we can say this that pair by sequence alignments are more suitable than the multiple sequence alignment, because we can calculate the maximum possibilities of highly conserved regions, and it is best then because it calculates only it is used for the two sequences. Thank you.